Hi everyone. We're going to start the Wednesday replacement recording session. We are moving on to the session 3 and 5, which is the inbreeding. Before we moving on to the session, I just recap the important things we have to discuss for your exam preparation because some students has some questions about this topic. So if you look at the how we represent the genetic variant or mutation from the database, we can represent the this information into three different formats. So one is the like we can use the DNA position, like a chromosome 2, certain position, and the G reference allele changed to variant allele, which is A, right? So this is the way we can represent the genetic variants on DNA sequence. Another way we can do that, we can represent the same genetic variants on the mRNA information, right? So in this case, we put the C dot. C is represent for cDNA made from mRNA, right? So this is the that information. And the position here is the different numbers compared to here, because this position is the nucleotide position in mRNA sequence rather than DNA sequence, right? So mRNA sequence of SN2A gene is composed 6,018 nucleotides in mRNA transcript. So out of these numbers, the mutation sitting in the 2,558th position, right? Mutation is not the 2,500 length, right? This is the position of the mRNA transcript. And again, G mutation changed to A, which is same as here. And if you look at the mutation, we are talking about the same mutation, right? So you need to remember that. So the same mutation, DNA scale, RNA scale. In this case, we are looking at the protein level. The mutation happened at the 853 position out of 2005 amino acid on protein. So these numbers are the number of protein, number of amino acid in protein, right? At which position we have this mutation is 153 position, right? And this position has the arginine, which is R here. And if the mutation happen, this arginine changed to glutamine, which is Q, right? And since this is the protein, so we put the protein P here, right? So P dot R changed to Q, the position in here, right? So essentially they are the same mutation, but we have the different notation for representing the genetic allele. Okay, so same concept. If you go to the samples of SN2A, we are talking about the wild type, which is the GG allele, right? And if the mutation happen, which is the containing the variant allele, it goes to T, right? So G changed to T. So this case, we have heterozygous genotype in this case, right? So one allele is G, another allele contains T, right? So we have the diploid, right? So one of the chromosome has the T. And then this G to T mutation that leads to the protein amino acid change from the glycine to stop codon, right? So this G is not here. So the same as this one. So this is the guanine and this is the glycine, right? So if you look at here, no mutation. So glycine, this position, same glycine. But here, if the mutation happened, then glycine changed to stop codon, right? And it leads to certain phenotype. So this is how you understand the concept of the mutation. Okay, so back to the inbreeding. So inbreeding is the concept we have to understand in the context of the population structure, because this is very obvious pattern we see in a natural population, including humans. Traditionally, inbreeding studied in the crop or animal science because the the performance of getting good product out of the breeding situation is really matter. So people talk about the inbreeding at the discipline, but nowadays as I mentioned many times, we have more and more genetic data set describing the human population. So we see very clear evidence about inbreeding in our population. So it's good to discuss this topic at the end of the population and mutation session. Okay, so we're going to learn about the three things. One is the inbreeding in human population and how we're going to measure the inbreeding, because anything we want to describe the genetic patterns or genetic phenomena, we have to come up with a certain measurement, right? So as we see in the previous slide, we talk about the fixation index, we talk about the what was that? The familial relationship or the linkage relationship, like a D prime concept. So inbreeding, we obviously has the certain measurement for that. And afterwards, we briefly maybe talk about the implication in inbreeding. So first, we're going to talk about whether inbreeding is really widely happening in human population or not. So we can find one note from Darwin's writing. So in 1838, Darwin proposed to his cousin Emma Wedgwood, and they were married in 39. And Darwin appears to have written these two notes weighing up the pros and cons about this marriage. Why he talking about the marriage? Because he's not a geeky person. He's more like a certain family history, which is he had a really happy marriage time, but and also he has 10 kids out of his time, but out of these 10, 3 dead because of his marriage, because Charles Darwin has very close marriage, which is his cousin. Then he thought that the cousin marriage, might occur some advantage in the offspring, because he thought that he cannot explain clearly, but he thought that there must be something disadvantage from the relative marriage given his life, right? And this is the familiar relationship of Charles Darwin's marriage. So here is Charles Darwin, and Emma Wedgwood is obviously his cousin. And this sort of relative marriage is quite common in human population, and surprisingly common, right? And in other words, we can say that non-random mating is common in human population. So in the history, we had a cousin marriage, sort of we see in the Charles Darwin's case, but also even modern days, we can think about like a physically or culturally, socially, we have certain preference. So we're not marrying with any random person, right? So we very has a specific preference and we want to marry with someone. So this sort of the situation we collectively call associative mating, right? So think about the situation, the Hardy-Weinberg equilibrium. What is that? The allele frequency has to be maintained, which is like a stable equilibrium across the generation. That's sort of the Hardy and Weinberg's assumption, but it can be violated because we not married with anyone. We had very specific mating, right? And this non-random mating is one of the major source to violate this equilibrium. Then this non-random mating can alter allele frequency over the generation, right? So that's why we care about this issue in the population structure. And on top of that, humans also has very small population size and also unlikely mate with the geographically distant organism in the past, right? So think about the situation like, 500 years ago, it's likely to meet someone in a different continent, right? Because we didn't have really good navigation skills, transportation. So we always married with someone geographically in the same region, right? But nowadays, a bit different. And the mating had been happened between close relatives. That also not a source of non-random mating. So this is sort of the distribution of marriage between couples, and can you see here on my Zoom? And as second cousins or close. So if you look at the percentage, the rates of consanguineous marriage across the countries, we can see a bit of different rates between the countries. So if you look at the Middle East, they have really high percentage of consanguineous marriage, around like a 50%, right? If you look at like a Western country, like a Canada, United States, Australia, Europe, they have around like a 10%, right? Quite high than your awareness, right? If you look at the Indian Peninsula, they have higher rates as well because they have many tribes and they married with each other. If you look at the Chinese, Malaysia, Japanese, they also bit of high rates as well. If you look at Korea, quite low. It's almost less than 1%, even close to zero. It's not meaning the missing data. We really low because we are the country, the ban the cousin marriage, even like a third degree cousin marriage is not allowed in our country over the half century, quite long time. So that's why it is quite lower percentage. So when we describe this relative marriage or close marriage, we can have several terms. One is the consanguinity, the mating among relatives, for example, first or second cousins. Literally, we call that this is the same blood. Endogamy is the marriage within a population or community, right? So if you look at the Middle East or Indian Peninsula, it is quite common. They have marriage within their tribes, within their community, was the same, social background. And compared to these two terms, we have different, concept, which is the cosmopolitan population. These are the population that are not isolated, right? So they typically urban population. So think about like a 21st century. We have a great advance in transportation. We move into different area. We're sitting there for education, like some of the students listening to lecture is from abroad, and the same situation. They go outside, they go abroad, and they find someone, and they may have some marriage at the place, and they compose the cosmopolitan population because they have a different genetic background, right? So this case, coming to modern society, we'll see more and more case of we can describe cosmopolitan population because these days geographically or social background is not the limited factors compared to previous generation. This is one example. We can talk about the cousin marriage in South Asia. So if you look at the Indian Peninsula, you can see the Indian area and the Pakistan area. And if you can see that there are many different tribes across different geographic region, and they have different religion. Like some people, they follow Hindi, some people follow Muslim, the Islam, and they have different background. So if you measure the genetic component, which is an allele and genetic variant, and if you draw the PCA onto that, then you can see quite distinct patterns, right? So if you look at here, so from here to here is the tribes we can get from the Indian Pakistan area, and these two is like a control group, right? So one is the CEU, which is the Central Europe. The other is the sorry, Chinese Han Beijing, right? So if you look at the European, they sitting around here, very distinctly separate from others, right? And other groups on here, these are the Korea, I guess, right? And these clusters are Saharia, right? So each tribes, they cluster themselves because, which means that they have very close marriage within a tribe, right? Rather than mixed each other. So they unlikely mixed marriage between this area, right? They marriage within their population, within their tribes, right? The population same. So if you look at the what was it? If you look at here, so this is the Chinese Han Beijing, right? And some of the Indian tribes they sitting around here, which is that the Nisha and Aonaga. And you can find the Aonaga here, Nishi here, and we can expect that the China around here, right? So they, geographically close to China, so they have the close, relationship in terms of genetic diversity, right? So this is again another example. So we can think about the population structure. Another way we can think about the inbreeding in our history is the consequence of genetic drift. So last time we talked about the Anabaptist population, like an Amish, Mennonite, who tried to migrate from Central Europe to United States. And they sitting there around like more than 200 years. And they have very distinct social background. So they keep their heritage within their religious community. And they have very distinct genetic pool, small genetic pool, and very isolated. And this is consequence to certain genetic disease. So you can see different prevalence given this effect. And we saw about the we discussed about the some inbred population, through the Indian population or like a cousin marriage and the Anabaptist. But we can also think about the opposite examples, right? So the random mating organisms. So there are organisms one example is an eel, right? So the fish. So this eel, they have random mating. They don't have associative mating. They don't have the non-random mating. They mating randomly, right? So this case we call this is a panmixia, right? We can also think about the panmixia examples in a natural population. Other than here, we can't really find a good example, especially in the animal kingdom, because the even animals they have associative mating, so they have the social structure and they have preference in terms of physical attractiveness. So even like a bird, even like a lions, they have different preference within their society. So this case we can also call associative mating. They're not the random mating. Okay, so when you measure these inbreeding patterns, we have certain measurements for that. So when you talk about the inbreeding, we have to refer coalescence theory, right? So what is the coalescence theory? Remember that? So when you describe the present population, so if you look at the three people here, if you want to describe an allele and how these alleles transmitted from the past generation. So if you track down them, you can see the common ancestor, passing these allele to the current generation. So the question is how these things pass from the past to here and how we can think about whether these allele are the same one as the common ancestor or not, right? That's important things. So if you want to figure out these questions, you have to come up with two different terms. So first one is identity by descendant, shortly we call IBD. Another one is identical by stage, shortly we call IBS. So let's start off the IBD first. So IBD is the thing, you have some parts of the chromosome in different individuals, right? So some parts means that like this, we can think haplotype, we can think genotype, we can think allele. So anything we can think about the genetic components on the chromosome, right? And if you look at the different individuals, like in here, are identical by states. So that's first thing. And second one is because they were inherited from common ancestor, right? Common ancestor is the second one. So if you look at here, if you look at this orange bar on here and orange bar here, right? So if you look at that, they are the same states, right? Because they are the orange one, right? And the second question is whether they are from the same ancestor or not, right? So if you go back there, you have here, and this one come from here, right? Come from here, come from here. And these things come from this person. So they are from the same ancestor as well. So this case we can call IBD, identity by descendant. So they are the same states, they're the same ancestor. Okay. So this is an important thing because quite many students every year has the question about this IBD concept in the heritability concept, right? So when you learn about the heritability, when we calculate the heritability between the parents and offspring, we calculate that 2 and 1 and the additive factors, right? And if you compare the offspring 1 to offspring 2, let's say two brothers, two sisters, brother, sisters, whatever, between the full SIBs, we calculate that additive, same as, parent and offspring relationship. Plus, we also consider dominant factors. And many of students' question was why parent and offspring not calculate these dominant factors. Why? Because if you look at the AB dominant genotype in a kids, let's say offspring 1, and if you compare with father has AB, okay? And where this first look is this AB and this AB, both dominant genotype, let's say, they are the same, right? They are the same, okay? Then the question is, this A comes from father, right? In offspring, and this B comes from mother, okay? But if you look at the father's one, this A comes from, let's assume that this is from grandfather, okay? So grandfather's A allele come to father, and grandfather's A allele comes to offspring. So this is essentially from father, but also from grandfather. And where this be from? This be from grandmother, okay? So this is different, right? So this B comes from grandmother, but this B on the kids, they're from mother, right? So mother's B from either grandfathers-in-law or grandmothers-in-law. So they're different. So even though they are the same states, but they are not from the same ancestor. Okay? It's very simple, right? So think about that. So we're not talking about just identical by states. We also have to deal with the same ancestor. That's the important things. So when we describe the IBD, we are always talking about the whether they have the same, ancestor or not, common ancestor or not. But if you want to describe only states, then we use the different word, which is the identical by states. So this is just merely asking whether alleles are from the same form or not, right? They look same or not, okay? So don't be confused between IBS and IBD. So this is another example. This ancestor has an G allele and somewhat has mutation happen here, and G changed to T. So T happened here and here, here and here. They have the same, they have the IBD situation, right? Because they have the same ancestor. Okay, so that's basically what we discussed. Also, IBD we call autozygous. IBS we also call allozygous. The same meaning. Okay. So when you measure, based on this information, we can measure the inbreeding coefficient, right? So inbreeding coefficient, in other words, we can refer probability that two alleles in multiple individuals will both descend from single allele in an ancestry. So this case we call identical by descendant, and also the probability that an individual is autozygous, right? This is one example. If you have the mating between, full sips, then your IBD is going to be 0.25 because it's the 0.5, 0.5, and it's going to be 0.25. And if you have the backcross, which is one of the parents with the offspring, then IBD also forming 0.25. This is an example. They making a full sim mating across many generation using Drosophila, obviously not using the humans, using the animals. So what this study wants to demonstrate, when you have more and more generation with the inbreeding mating, the full sim mating, you will see the higher inbreeding coefficients after many generations. Yes or not? And when you measure that experiment, that is actually yes, right? So when you have the full sim mating and a one generation, inbreeding coefficient was 0.1. But then after generation, second and third, fourth, fifth, then inbreeding coefficient gets higher. So this is clearly, we can see that the inbreeding happen, then the inbreeding coefficient working as expected. And another way we can measure this inbreeding coefficient using genetic data set. That's more important in these days, right? So because we can directly measure genetic composition in each individual. So one way we can do that is we measure the heterozygosity. So if we measure the 10 people in a population, let's just say population 1, and this is population 2 and population 3. If you measure the heterozygous variant, how many people has heterozygous? So some people can have homozygous variant, which is large AA, and some people has the heterozygous variant, which is the large A, small a, and 50% people has the heterozygosity, and second population 20%, third population 0%. So what you can get conclusion from here, population 1 has more diversity, and population 3 no diversity, and they might experience the inbreeding situation, right? Because they have only one genotype. So extending this idea, we can think about sort of the time flow, right? So this is the examples of loss of heterozygosity. So this is very old time, and this is tens of human. And very early initial phase, we have the mixed of certain genotype, right? So one genotype, some people has homozygous, some people heterozygous, very different heterozygosity at the time. And after 6 generation, so let's say one generation takes 20 years, so 20 or 30 years. So after 6 generation, it's about like 150 years, right? And that time, after a century, few people, about 6 people, they have, oh, sorry, not 6 people, 3 people, because this is the diploid. Sorry. Okay, anyway, so 3 people, they experience the genetic drift, right? So only small people subset out of the population, and they have one generation, right? So this is an example. This is the situation like, some people and only few people, they migrate to different continents, like a Anabaptist, or they migrate to United States, like the Finland people or out of Africa, and they compose the new population from here, right? And they start from the three people, and they have the offspring, they have a new generation, and after some generations, only few people also migrate again to another place, and they constitute the current population. And you see that the lower genetic diversity leads to high level of homozygous variants, which means that heterozygosity is disappeared and homozygosity is up there, right? So this is sort of very schematic plot, but this is recap the inbreeding patterns we discussed so far, right? And also one more thing you have to remember is when we describe the Hardy-Weinberg equilibrium, we thought about the Punnett lecture, right? So brachytidal is the dominant allele, but if you look at the frequency of brachytidal in the population, they're very low, right? Even though they are the dominant allele, right? So that was the questions, driving the Hardy-Weinberg equilibrium, right? So the question is how this allele frequency is determined at a very early days, right? This is the answer we can think about here, right? So few people migrate to certain area, they forming a new population, right? So think about the situation like out of Africa, from the Africa, only few people, they migrate to Europe, they migrate to Asia, they migrate to America, and they forming a new allele frequency. Does that make sense? Right? So don't think about like a current situation. We have the, full of people in the Europe, in the Asia, in the other continents. We're not talking about the current generation. We're talking about like 100 years ago, 100,000 years ago, right? So only few people, I don't know how many people. It won't be like a more than 10,000. It will be like you know, tens of people. They migrate to Europe, they migrate to Asia, they settling down. And those handful of people, they determining the allele frequency in a new population, right? So this is how the allele frequency determined at the first time, right? So that's why either dominant allele or recessive allele does not really matter in the allele frequency, because allele frequency is set by initial populations rather than either it's dominant or recessive. That's the important things you have to keep in mind. Okay, so this is the flow of loss of heterozygosity. So in other angle, we can think about inbreeding in the DNA data, right? So the previous slide, we talked about the heterozygosity, but this time we talk about the homozygosity, right? So this is the measure homozygosity level in a population. So which means that this is sort of the counterpart of the heterozygosity, right? So higher heterozygosity is the less inbreeding, but higher homozygosity is the higher inbreeding, right? So how we can measure that? So this is the examples of chromosome 2. So if you look at the chromosome 2, very first position to 250 million base pair, you can plot the variants across the positions, right? So this is very, very lower resolution, so you cannot see distinct representation of each variant because it looks like overlap to each other, but it actually the first position, second position, they have the different genotype in every locus, right? So you have to make sure that. Okay, because it's squeezed, right? So how we can measure that? You can basically measure either it's a reference genotype or heterozygous genotype or homozygous, variant homozygous genotype. Then we can measure that by B allele frequency. please make sure that this is not the same allele frequency we discussed in a population level. This is only, used in the individual level, right? So how we can measure that? You remember that we talked about the depth last time, right? So when you measure the genome sequencing, you can have 4x, which is you read the DNA 4 times. You read the DNA 1 time, 2 time, and 4 time, we say it's the 4x, right? So if you read the 4 times at this position, then you have all 4 possible counts. Then how many times you see a small a allele at the position. So if you read the 4 times, if you see 4 times, then it will be 1. That is the BAF, B allele frequency is a 1. Because at the position, you only have small a there, right? So we can easily infer that that position has small and small a, variant homozygous genotype. But if you read the 4 times, if you have only 2, then that's going to be 0.5. Why? Because 2 times it will have the small a, 2 times it will have the large a. So that's why it's going to be 0.5. And if you read the 4 times, and if you have only 0 times have small a, which means that another 4 only comes from the large a. So that's going to be 0, okay? So using that information, you can infer that either it has the small a, or heterozygous, or the large A, right? And this data is not obviously read 4 times. It would more than 4. It probably like 100. How we can know that? Because if you read 4, your data point only be 1, 0.75, 0.5, 0.25, and 0. But it won't be like that because it has some point is between this range. So let's assume that they read like 100 times, right? And some of the locus has the BLE frequency like a 0.8 or 0.7 something. That's likely be homozygous, variant homozygous genotype or heterozygous genotype. We can really tell them. Why? You have to remember that this is an experimental data, so it's not exactly happening as you expected. Some of the measurements by experiments can be noise, right? So this can be noise, right? This can be noise. This can be noise, right? So your expectation either be here, but it can be noise, right? So that's why we have a bit of off numbers there. But anyway, so we measure the BAF, then we can measure that the locus, they randomly shuffle between the 3 genotypes, right? But if you look at this person, it turns out to be yes, but this person was not. Why? Because some of the regions like in here and here and here, they're missing. Why? Because this area has only either be variant homozygous genotype or reference homozygous genotype. Only homozygous genotype. There is only homozygous genotype, only homozygous genotype. This case, we call this is the runs of homozygosity area, which is the ROH, right? Why these things happen? Because this person's parents, mother and father, they're genetically close. So they have very similar genotype within this area. When they marry and when they mate and they have offspring, they have the same genetic blocks and they're forming the homozygosity block in here, which is in ROH. So that's why we see ROH blocks on this person. Okay, so that was the concept of ROH. And one more thing I need to mention is you can see this area is missing, right? And this area is missing. Why? Because this area is the centromere. So we cannot have the DNA sequence in that position. Okay. Okay, let's move on to here. So if you look at the ROH in human genome, you can expect the size is around 500 kilobase to 1 million base pair size. And as we see before, it's an absence of heterozygous allele. And this is the indication of level of inbreeding in a population, right? So when you see some person from inbred population, your expectation is that person might have higher, level of ROH compared to, cosmopolitan population. So we can use this information to infer how far the inbreeding happened in a population. Okay, so now we have the inbreeding coefficient, we have the heterozygosity, we have the Russell homozygosity, and we can see that how these things happen in a real human data set. This is the paper from Pemberton and Rosenberg in 2014. I really love this paper. And they measured the different population from Africa to Middle East, Europe, and Oceania, America, and Latino. And they used the microarray and they measured the heterozygosity within each population. First look, what you can tell is African population, they have higher heterozygosity level compared to non-African population, which is including all of them, right? Why? Because as I mentioned previously, out of Africa, they experience small sets of people, they migrate from Africa to other continents. So, which means that small population, genetic drift, they experience very low genetic diversity compared to original population. And original population is an African. African has really higher genetic diversity, and they obviously has the higher heterozygosity level. And compared to African, non-African population, they have lower, especially some people from Oceania, some people from Native American, they have lower heterozygosity because they have consanguineous marriage. They're geographically isolated and they based on the tribes. So that's why it's reflected there. This is another paper measuring different concept, which is the runs-off homozygosity. So they measured how much we have runs-off homozygosity in our genome. So they measure the proportion and they put in here F and FRH, which is the fraction, right? So they compare the African, which is red, and they compare the Middle East, which is the purple. They compare the Europe, which is green. They compare the East Asian, which is purple, which is blue, sorry. And if you look at that, first look is the African population. They have the lower homozygosity fraction, right? So this is the sum of homozygosity. So from 100 to 600 megabase pairs, right? So given 3 billion base pairs in our genome, like in East Asian, we have 500 million base pair, which is around, 1 in 6, around like 50%, 15%, right? Quite high than your expectation, I guess. But if you look at the African, they only have like less than 10%. They much more heterozygous genotypes in their DNA, right? So if you look at that, the African versus non-African, we see clear distance, right? And non-African population, they generally high, but especially the Middle East quite high, and East Asian is high, right? And if you look at the length distribution, so in each blocks, how many counts you can expect from each length intervals? Africans, they very short, and they have less numbers. And Asian, they short, they many numbers. European, they short, many numbers of the ROH blocks. Middle East, they have many numbers. They short, but they have super long ROH block. Why? Because they have consanguineous marriage. They have very high level of that. So they likely have genetically same parents. So that's why they have the larger genetic block. Not only they have the more ROH proportion, they also have very large block for that. Okay, so this is the sort of the part of entire lecture. So if you can make a conceptualization about the RH relationship between the different population size and population type, we can summarize into like this. So if you look at the population, large population like in African, the sum of the RH, the sum of RH is like this level, and the number of RH, this level, y-axis. If you compare the large population to small population, let's assume that this is European, and sum is increased, right, compared to here, and the numbers also increased as well, right? But if you look at the admixture population, or we can say admixed, which is European ancestry married with the Asian ancestry. Does that case, they have genetically very different. So RH is getting lower, and the number of RH getting lower as well. If you have consanguineous marriage, as we see in the Middle East, they experience the longer RH blocks, right? So they have the total RH proportion increased, even though they have the same numbers, because they can have the longer RH block. If you experience a bottleneck, then it will be increased than the small population because they much smaller, people by the drift event, right? So they increase the sum of RH, also number of RH as well. And they also increase the RH length as well. Okay, so that's sort of very important concept. We have to think about the RH relationship. So what is the implication we can think about the inbreeding? So first thing is the functional consequence of the inbreeding. So Charles Darwin's question in 19th century was cross-fertilization. So he wanted to know about the advantage between the cross-fertilization versus self-fertilization. So self-fertilization basically meaning the inbreeding situation, and cross-fertilization is less inbreeding situation than that because different genetic components, right? So his experiment is he excluded potential insect pollinators. So Darwin conducted the control cross and self-pollination because the insects can, mediate some, cross-fertilization between the geographically different areas. So he controlled that experiment. So he used 57 species from 52 genera and 30 families from the certain plant. Then he compared the different, the offspring between the cross and self-pollination. Then he continued that experience over 11 years, including 10 generations, self and cross-pollination. And what he found was the vast majority of cases, cross-pollinated offspring, they have better performance than self-pollinating offspring. So compare the length of the plant or how many numbers plants can have like a fruit or something like that. So all of that, the characteristics when he compared that, so cross fertilization is better than the self-fertilization. This is indicating the inbreeding is unfavorable to the survivors and also the performance in terms of the phenotypic appearance. So this case leading to other discussions when people talk about inbreeding, they thought that inbreeding might be unfavorable and way lower performance in the natural selection. So this case we collectively call inbreeding depression, right? So mating between close relatives has detrimental consequence on the survival and fertility of resulting offspring, right? And And this is also examples from Charles Darwin's experiments. And inbreeding depression is important topic when they talk about the evolution and the mating system. And one of the things very commonly discussed in the crop science is the heterosis, right? So if you have the homozygous parents, like inbreeding A parents inbreeding B parents, if you mix them, the offspring has way outperformed phenotype than these two parents. In this case, we call the heterosis. So heterozygous genotypes way better phenotypic manifestation than the inbreeding parents. And this is also important for the crop science, even in these days. So people keep talking about this topic. But what I want to bring this topic to you in this session is, the inbreeding is one of the major characteristics of population structure. So that's why I'm giving you some thoughts on that. Another one is inbreeding depression is widely talking about in animal and crop science, but I guess this will be very interesting topic in a human genetics in your days. So some pieces of information we can get from recent studies. So traditionally we can think about this inbreeding depression in a recessive disorders in consanguineous marriage, right? That is very common topics. But recent years we have the biobank data set, so people measure that one into very systematic way. So they measure many different phenotypes and which phenotypes they are affected by inbreeding patterns, so they can measure mathematically standardized. And also they have some of the information in the polygenic situations as well. And also very interesting study come from UK Biobank, which is they're looking at the Pakistani heritage UK Biobank resident, right? So some UK people, UK residents in the British society, they're the immigrant, and one immigrant has really distinct social culture community in the England. So I guess this is from the Bradford, I guess. So that area has really huge community of the Pakistani immigrants. And some people from the Oxford, they measure their genome and they measure whether inbreeding, high level of inbreeding, impact on their developmental issues or not. And they found some interesting pattern from these papers. And sort of this idea, I think, is sort of ongoing efforts to looking at the the isolated communities or tribes and whether the health conditions can be described by this inbred related genetic components. I guess this would be continuing interesting topic in a near future genetic studies, I guess. Okay, so we learned about the inbreeding, and we ended the session for population imitation, and this is the sort of range we're going to do a midterm exam. So from the session 1 to session 3. Session 1 is just an introduction though. Mostly session 2 and session 3. So if you have any question, it's always welcome to send me an email and I can, reply to you and also post on the Notion so everyone can see that and also make a continuing discussion. And also we're going to have the Q&A session next week, Monday and Wednesday. So if you have the questions, feel free to bring your one to the question time. I highly encourage. And then we don't have the formal class next week. We Q&A session, so for your reference. Okay, see you next time.